cell growth, division, and reproduction. Limits to cell size. The larger a cell becomes, the more demands the cell places on its DNA. In addition, a larger cell is less efficient in moving nutrients and waste materials across the cell membrane. There are two main reasons why cells divide. Information overload. As a cell grows, that information is used to build the molecules needed for cell growth. But as a cell increases in size, its DNA does not. If a cell were to grow too large, an information crisis will occur. Exchanging materials. Food, oxygen, and water enter the cell through its cell membrane. Waste products leave a cell in the same way. The rate at which this exchange takes place depends on the surface area of the cell, which is the total area of the cell membrane. The rate at which food and oxygen are used up and waste products produced depends on the cell's volume. Ratio of surface area to volume. As the length of the sites increases, the volume increases more than the surface area. Division of the cell. Before it becomes too large, a growing cell divides, forming two daughter cells. This is a process called cell division. Cell division and reproduction. Asexual reproduction. The production of genetically identical offspring from a single parent is known as asexual reproduction. The process can be simple, efficient, and effective, enabling populations to increase in number very quickly. Sexual reproduction. Offspring produced by sexual reproduction inherit some of their genetic information from each parent. It involves the fusion of two separate parent cells. For single-celled organisms, asexual reproduction is a survival strategy. When conditions are right, the faster they reproduce, the better their chance of survival over other organisms using the same resources. Having offspring that are genetically identical is also an advantage as long as conditions remain favorable. However, a lack of genetic diversity becomes a disadvantage when conditions change in ways that do not fit the characteristics of an organism. Sexual reproduction is a different type of survival strategy. The process of finding a mate and the growth and development of offspring require more time. However, this can be an advantage for species that live in environments where seasonal changes affect weather conditions and food availability. It also provides genetic diversity. The process of cell division. Chromosomes. Cells can handle very large molecules only by careful packaging. Genetic information is bundled into packages of DNA known as chromosomes. Prokaryotic chromosomes. Prokaryotes lack nuclei. Their DNA molecules are found in cytoplasm. In most prokaryotes, a single chromosome holds most of the organism's DNA. Eukaryotic chromosomes. They contain multiple chromosomes that form a close association with histones, a type of protein that is referred to as chromatin. As a eukaryotic cell prepares for division, each chromosome coils more and more tightly to form a compact structure. Chromosomes make it possible to separate DNA precisely during cell division. The cell cycle. During the cell cycle, a cell grows, prepares for division, and divides to form two daughter cells. The prokaryotic cell cycle. Their process of cell division is in the form of asexual reproduction, known as binary fission, that is, cell division in a single-cell organism that produces two genetically identical organisms. Then the eukaryotic cell cycle. This cycle contains four phases, G1, S, G2, and M. Its length varies depending on the type of cell. The time between cell division is called interphase and is divided into three parts, G1, S, and G2. G1 phase, cell growth. Cells increase in size and synthesize new proteins and organelles. S phase, DNA replication. New DNA is synthesized when the chromosomes are replicated. G2 phase, preparing for cell division. Many of the organelles and molecules required for cell division are produced. M phase, cell division. It produces two daughter cells. This is the process of mitosis. In eukaryotes, cell division occurs in two main stages, mitosis and cytokinesis. Mitosis, prophase, the first phase of mitosis. The chromatin condenses into chromosomes, the centrioles separate, and a spindle begins to form. The nuclear envelope breaks down. Metaphase, the second phase of mitosis. The chromosomes line up across the center of the cell. Each chromosome is connected to spindle fibers at its centromere. Anaphase, the third phase of mitosis. The sister chromatids separate into individual chromosomes and move along spindle fibers to opposite ends of the cell. Telophase, the fourth and final phase of mitosis. The chromosomes which were distinct and condensed begin to spread out into a tangle of chromatin. Cytokinesis. It completes the process of cell division. It splits one cell into two. It usually occurs at the same time as telophase. Cytokinesis in animal cells. The membrane draws inward. Cytokinesis in plant cells. A cell plate forms. Point 3. Regulating the cell cycle. Biologists discovered a protein in cells that were in mitosis. When they injected the protein into a non-dividing cell, a mitotic spindle would form. They named this protein cyclin because it seemed to regulate the cell cycle. Regulatory proteins. The cell cycle is controlled by regulatory proteins both inside and outside the cell. Internal regulators. Internal regulatory proteins allow the cell cycle to proceed only when certain events have occurred in the cell itself. External regulators. External regulatory proteins direct cells to speed up or slow down the cell cycle. One important group of external regulatory proteins is the group made up of the growth factors. Growth factors stimulate the growth and division of cells. Apoptosis. It's a process of programmed cell death. It plays a key role in development by shaping the structure of tissues and organs in plants and animals. Cancer. Uncontrolled cell growth. Cancer is a disorder in which body cells lose the ability to control growth. Cancer cells do not respond to the signals that regulate the growth of most cells. As a result, the cells divide uncontrollably. Cancer cells form a mass of cells called a tumor. Cancers are caused by defects in the genes that regulate cell growth and cell division. The control over the cell cycle has broken down. An astonishing number of cancer cells have a defect in a gene called p53. These genes cause cells to lose the information needed to respond to signals that normally control their growth. Treatment for cancer. A cancerous tumor can be removed by surgery. Many tumors can also be treated with radiation. Chemotherapies can also be used, but they cause serious effects on patients.
10.4 Cell differentiation. All living organisms start out as one cell. They pass through a developmental stage called an embryo, from which the adult organism is gradually produced. During this process, the cells begin to differentiate and specialize for particular functions. The process of differentiation determines a cell's ultimate identity, such as whether it will spend its life as a nerve cell or a muscle cell. Stem cells are unspecialized cells from which differentiated cells develop. Stem cell research provides many benefits, such as possibly developing a new field of regenerative medicine and more. However, it also presents many ethical issues, as the most potent form of stem cell can only be harvested from unborn embryos, and all the current methods leave the embryo dead. 11.1 The work of Gregor Mendel. Genetics is the study of heredity, which is key to understanding what makes each organism unique, and the modern study of genetics was founded by an Austrian monk by the name of Gregor Mendel. After becoming a priest, Mendel spent much of his time in a monastery, where he was in charge of the monastery garden, where he performed many scientific experiments. His most famous experiment involved pea plants, as they were easy to grow and also produced hundreds of offspring. Mendel's discoveries led to the conclusion that every trait will be passed down from parent to offspring. These traits are called genes. The different forms of each gene were called an allele. Mendel's discoveries also led to the conclusion that some alleles were dominant and others recessive, where the dominant allele will always be exhibited.
Applying Mendel's principle, organisms that have two identical alleles for a particular gene, capital T or lowercase tt, in this example are said to be homozygous. Organisms that have two different alleles for the same gene, such as capital T and lowercase t, are heterozygous. All reason for observation of the F1 crosses is that every organism has a genetic makeup as well as the observable characteristic. All of the top pea plants have the same genotype or genetic makeup. However, they do not have the same genotype or genetic makeup. The genotype for an organism is inherited, and the phenotype is largely determined by the genotype. Phenotype can be reminded as what we see, and the physical, and the genotype is what we cannot see. Punnett square uses mathematical probability to help to predict the genotype and the phenotype combination in genetic crosses. Mendel wanted to discover even more. An example of this is of his many wonders is that he wondered the segregation of one pair of alleles affects the other one. He followed the characteristic in different generations. The two-factor cross F1 plants were heterozygous for both seed shape and seed color. The two-factor cross F2, the allele for seed shape segregated independently for who does for seed color. Genes that segregate independently do not influence each other's inheritance. Mendel had discovered the principle of independent assortment. It states that genes from different traits can segregate independently during the formation of gametes. Mendel's principles are the inheritance is determined by individual units called genes. In most sexually producing organisms, each adult has two copies of each gene and one for each parent. With all the investigation of Mendel, more investigations were done about genetics. Other patterns of inheritance. 11.3 Incomplete dominance is where one allele is not completely dominant over another. A great example of this is on the flowers. A generation produced by a cross of white colored flower and a red colored flower, the result of this one is a pink flower. Neither had complete dominance over the phenotype. Codominance is where both alleles are clearly expressed. An example of this will be the chicken that is brown with black points on their pattern. Many genes exist in several different forms and therefore said to have multiple alleles. And it is a gene that has more than two alleles. Many traits are produced by the interaction of several genes. traits controlled by two or more genes are said to be polygenic traits, and it means many genes. Environmental conditions can affect the gene expression and influence genetically determined traits. Meiosis. A diploid cell is a cell that contains two sets of chromosomes. This is double the haploid chromosome number. Each pair of chromosomes in a diploid set is considered to be one homologous chromosome set. Haploid describes a cell that contains a single set of chromosomes. Mitosis is a type of cell division that results in two daughter cells, each having the same number and kind of chromosomes as the parent nucleus, typical of ordinary tissue growth. Mitosis consists of four basic phases: prophase, metaphase, anaphase, and telophase. In early prophase, the cell starts to break down some structures and build up others up, setting the stage for division of the chromosomes. The chromosomes start to condense, making them easier to pull apart later on. The mitotic spindle seems to form. In late prophase, the mitotic spindle begins to capture and organize the chromosomes. The chromosomes finish condensing, so they are very compact. The nuclear envelope breaks down, reducing the chromosomes. In metaphase, the spindle has captured all the chromosomes and lined up at the middle of the cell, ready to divide. In anaphase, the sister chromatids separate from each other and are pulled toward the opposite of the ends of the cell. In telophase, the cell is nearly done dividing, and it starts to reestablish its normal structure as cytokinesis, division of the cell contents, takes place. Cytokinesis is the division of the cytoplasm to form two new cells, overlaps with the final stage of mitosis, and it may start after the anaphase or telophase, depending on the cell, and finishes shortly after the telophase, and the result is two daughter cells. During meiosis II, the sister chromosomes within the two daughter cells separate, forming four new haploid gametes. The mechanics of meiosis is so similar to mitosis, except that each having cell has only one set of homologous chromosomes. Mitosis results in two identical daughter cells, whereas meiosis results in four sex cells.
Identifying the substance of genes. By observing bacterial transformation, Avery and other scientists discovered that the nucleic acid DNA stores and transmits genetic information from one generation of bacteria to the next. Hershey and Chase experiment with bacteriophages confirmed Avery's result, convincing many scientists that DNA was the genetic material found in genes, not just in viruses and bacteria, but in all living cells. The DNA that make up genes must be capable of storing, copying, and transmitting the genetic information in a cell. The structure of DNA. DNA is a nucleic acid made of nucleotides joined into long strands or chains by covalent bonds. The clues in Franklin's X-ray patterns enabled Watson and Crick to build a model that explained the specific structure and properties of DNA. The double helix model explains Charpat's rule of base pairing and how the two strands of DNA are held together. DNA replication. DNA polymerase is an enzyme that joins individual nucleotides to produce a new strand of DNA. Replication is that most prokaryotic cells start from a single point and proceeds in two directions until an entire chromosome is copied. In a periodic cell, replication may begin at dozens or even hundreds of places on the DNA molecule, proceeding in both directions until each chromosome is completely copied. RNA. The main difference between RNA and DNA are that the sugar in RNA is ribosed instead of deoxyribose. RNA is generally single-stranded, not double-stranded, and RNA contains uracil in place of thymine. In transcription, segments of DNA serve as templates to produce complementary RNA strand molecules. Ribosome and protein synthesis. The genetic code is read 3 letters at a time, so that each word is 3 bases long and corresponds to a single amino acid. Ribosomes use the sequence of codons in mRNA to assemble amino acids into polypeptide chains. The central dogma of molecular biology is that information is transferred from DNA to RNA to protein. Mutations. Mutations are heritable changes in genetic information. The effects of mutations on genes vary widely. Some have little or no effect, some produce beneficial variation, some negative disrupt gene function. Mutations often produce proteins with new or altered functions that can be useful to organisms in different or changing environments.